Okay, thank you. And I would like to thank first the organizer for inviting me to give this talk and show you some of the results that we have in our lab. Today I want to show you what we are doing to try to identify new molecules, but really they are not new. So they are all molecules, but with a new effects that in the early stage in a neural dysfunction. We have been working for maybe about 25 years in Huntington disease. And in the beginning, but we spent a lot of years trying to block the neuronal death. Maybe this was a mistake or something like that, because what could be useful is to go to the early stages and try to block or try to repair the neuronal dysfunction. So this is what we are doing here with the neuronal death versus neuronal plasticity. In the last year, we focused to try to identify the mechanisms that modulate the neuronal plasticity and neuronal dysfunction and try to find treatments for that. Well, you know that in Huntington disease, mutant Huntington is acting through so many different mechanisms in synaptic dysfunction, gene transcription, intracellular transport, mitochondria, autophagy, etc., etc. But we want to focus what we have been working with the trophic factors. With the trophic factors, we have been characterized the different neurotrophic factors that act on the different neuronal populations that are affected in Huntington disease, and really think that everybody agree that BDNF could be a very potent trophic factor for these molecules. BDNF can induce improve the synaptic function, the neuronal survival, and motor coordination, but it's also affecting the cognitive processes. And really, mutant Huntington is able to modulate the synthesis of BDNF, and also there is a reduction of BDNF. You can see here that BDNF is the most potent. So you see this is a wild type. It's a culture, striatal neurons, and when you treat the cells with BDNF, there is an increase in the branching, in the neural outgrowth, and also the survival is the most potent. There is not any other trophic factor with these clear effects. 10 years ago, or more than 10 years ago, Lena's Cataneo group described that it was a reduction in the BDNF levels in several mouse models, but also in human patients. So this was the beginning. Together at this time, we also found that in different animal models, BDNF could be protective in the excitotoxic effects. Sorry. Well, we did a paper sorry, that this effect of BDNF was really important for the pathophysiology of Huntington disease. And what we did is to develop a mouse that they have mutant Huntington, but low levels of BDNF. So we cross-made the R61 mice together with heterozygote for BDNF. And here it was, they developed severe symptoms in the motor coordination in the rotor rod. And I skip this. And also we could see that the population that was most affected was the encephalinergic neurons. This population they are the first ones that are affected in Huntington disease. So these papers, this study really show that BDNF has a role in the pathogenesis in Huntington disease. If we, in another set of experiments, we try to overexpress BDNF in a mouse model of Huntington disease, and what we did is we have one mice that only overexpress BDNF under the GFAP promoter. So in these mice, they have normal levels of BDNF, but when there is any kind of pathology, cytotoxic insult, there is an increase of BDNF. What we did is to crossmate with these animals with these R62 mice, and what we got is a double mice that have mutant Huntington, but overexpress BDNF when there is a pathology. Otherwise, it's normal. Here you see that this mice, you see that this is a mice with a mutant Huntington, and they have mutant Huntington, but with higher levels of BDNF, so that we could compare two animals with a mutant Huntington and the same number of repeats, but they have different levels of BDNF. In the previous one, they had the low levels, and this one we have high levels. And this was a protective effect. This is in the clasping, but also in the rotor rod, we could see that was an improvement in these mice because they have higher levels of BDNF. So this was another evidence that BDNF can modulate and the onset and also the severity of the symptoms. And here the postsynaptic and presynaptic markers were higher. So these studies and some other studies show that BDNF was involved, but we could check also about the receptors. How are the receptors that binds BDNF? BDNF binds two different receptors with a different function. One is the TrkB that induces survival, axonal growth, and also cell proliferation. And there is another receptor that is P75. This is not only for BDNF, could also bind other neurotrophins, and they have the opposite effect. P75 is a cell death receptor and can induce an inhibition of the axonal growth. So this is the situation that we have with a BDNF that can bind two different receptors with a different function. One is inducing as a negative modulator, and the other is a positive modulator of the synaptic plasticity. And how are they in Huntington disease? These are two papers that Silvia Ginés did and Veronica Brito, and what we found here is that P75 is increased, and also Lena's Cataneo group also found the same results in different mouse models and also in human. And we could see that P75 is increased in Huntington disease, in mouse models and also in human patients. And TrkB is decreased. These are the differences. So there is an imbalance between P75 and TrkB. To find the functional effect of this upregulation of P75, what we did is to develop a mouse that we cross. This is the knock-in mice with heterozygote for P75. So what we did is to decrease the levels of P75, and when we decrease the levels of P75, we could find and recovery of the spatial and non-spatial memory deficits, and also we improve the dendritic spines that were downregulated in the knock-in mice. So we could find. So that means that P75 is also quite important to modulate cognitive and also the number of spines. So I mean synaptic plasticity. So if we know that BDNF could be relevant and also the activity of its receptors, And the next step will be to put BDNF in the patients or on the clinical trials. And the main problem is the way that we can do that. So BDNF is quite large. There is poor pharmacokinetics properties, and also, I mean, it doesn't cross the blood-brain barrier. So we have to identify new ways to deliver BDNF to the brain, and the best one could be to try to modulate endogenous BDNF and also the P75 tract B imbalance. So even with exercise, there are some papers that show that BDNF can be increased, and good food and exercise can improve. But anyway, it's not enough, so we have to find another ways to do that. There are different papers that they try to do this pharmacological manipulation. We participated years ago in a paper with, we used cysteamine. We did this paper with a French group, with Sandrine Humbert and Frédéric Sardou. And this study opened a clinical trial to try to use cysteamine that was usually, it was used for different diseases, and it was then used in Huntington disease clinical trial. And what I want to show you today is there are 3 drugs that we have been studying during these last years, and with different effects. They are all of them are focused in the BDNF and tract B and P75 imbalance. And the first one could be the prostaglandins. And what we'll try to do is to stimulate endogenous BDNF. And the second one could be the hydroxyflavone, that to see if it's a real partial tract B agonist. And fingolimod, that it could be a modulator of the P75 and tract B imbalance. Okay, this is the first one. This is a prostaglandin signal transduction pathway that prostaglandins, as you know, has a lot of different activity, even in the vascular system, but they have receptors also in the brain. And there are different kind of receptors with different signal transduction pathways. And what we show you today is the EP2. So we started in a previous study that we did with EP1, and later on we did with EP2. That is what I want to show you today. This is the first study that really linked prostaglandins also with the pathophysiology of Huntington disease. And this receptor usually have a toxic effect in the brain. So what we did is to use an antagonist and try to improve the motor and also memory deficits in these mice. But if we go to the EP2, this the first point that why we use it is because the receptors are located in the brain areas that are really related with Huntington disease, the cerebral cortex, striatum, and hippocampus. And and there are also other papers that show that EP2 could be related with the secretion of BDNF. In these different papers, in different cultures, what they did is to find that prostaglandins can induce BDNF and also NGF, and also is this related with long-term synaptic plasticity. And then what we did is to use an agonist of the EP2 receptor, and we could find that when we treat the cultures, there is an increase in the, in the number of neurites. We use a misoprostol. I don't know if you can see here the slide. And also another interesting point, and that it was an increase in the release of BDNF after the treatment. This effect in the culture was BDNF dependent, because when we use an antagonist of, an antibody that blocked the BDNF effect, the effect of the misoprostol was also blocked. So that means that this prostaglandin receptor activation, what induces is an increase of BDNF dependent effect. If we move to in vivo, we could see that this study was done, now is to study cognitive effect, to study memory. So what we did is to study mainly in the hippocampus. This was interesting because this was a specific effect most in the hippocampus. This was not effect in the striatum and in motor coordination. When we measured with a T-maze or with a novel object recognition test, the NORD, we could see an increase and a recovery of the effect in the long-term memory after the treatment. We use a mini PAMS in these studies. These receptors are mainly affecting, or they have many receptors in the astrocytes, and we try to check if there is the markers of inflammation were also increased, but we could not see it. So there is an increase in BDNF, but in the other markers of astrogliosis or inflammation was not increased. So it seems it was more an effect specific of BDNF. When we did the effect in the presynaptic or postsynaptic markers, we could also be a recovery in these markers. Another interesting point is that there is a reduction in the effect of in the inclusions of Huntington. Well, this is the summary of the results that we got, and we could see that it's known that TP2 can induce through cyclic AMP and PKA to an increase in the phospho-CREB, and also this could be induced a BDNF release that also can have the regulation in the neuronal plasticity. Okay, let's go to the second one. This is a dihydroxyflavone. This is a drug that can be taken by a plant. And what we did is, it was a paper that showed that, described that this could be a good agonist, a potent agonist with an neurotrophic effect. And even what we also try is to identify the structure of this drug and to compare with BDNF and try to identify if we can make new drugs using the structure. And this is what they did in the beginning, but there was some problems that the previous results could not be reproduced for all the people. And then we spent a lot of time trying to see this really was an agonist of TrkB, or maybe it was another mechanism that has protective effects. You see here there is not any band to try to phosphorylate TrkB. But after a waste of time making so many experiments, what we found is the problem was the antibody. The antibody, there is a different residue that in the TrkB receptor to phosphorylate, and this is the 515 and the 816. And the main problem, what we could find in the next experiment, is that the hydroxyflavone only activates this part of the TrkB receptor. So finally, we could identify, and also this is interesting because this activates the PLC gamma pathway and not the other pathway. So what we found at the end was that this was a specific agonist of this 816 residue of the TrkB receptor. So here we could see that there is a quite good activation of the 816 and not of the 515. With the Western blot, we could see also that this activation of the PLC, the PLC gamma. After the treatment with this drug, we could see an increase in the branching of the neurites. And also, we could find, when we did the treatments in vivo, that there is an improvement of the motor activity, motor coordination. This is mainly in the higher speed, and also with the novel object recognition test. So there is an improvement also in the cognition deficit. What is interesting with this molecule also is the better effect were found in these encephalinergic neurons and not with the substance P. So this is more similar one that we could find in Huntington disease. These are in R61 mice. And also there is a reduction in the number of Huntington aggregates. So the results may mean that we have a drug that could be a specific agonist of one specific residue of the TrkB and could phosphorylate only the PLC gamma receptor. And just to finish, this is the fingolimod, that this is a drug that is used for multiple sclerosis. So this is approved by the FDA. And this is an agonist of the sphingosine 1-phosphatase receptor and is a modulator of the immune system. And we found that this paper, that really fingolimod could be also useful in a mouse model of Rett syndrome, and we tried to check if this could be useful for Huntington disease. And then we did this treatment. This is a long treatment with R61 mice. And we checked this study. What we did is the cognitive effect. There was also a previous paper that showed some results with a motor coordination. And we did here with cognitive effects in memory during the T-maze and the North. And the effect was quite clear with objective with a novel object recognition test, and also with the T-maze, there is a recovery after the treatment. Also with the number of spines, we check in the CA1 pyramidal neurons, and there was an improvement in the number of spines, and also with the PSD95 as a postsynaptic marker. Also, fingolimod is known that can modulate astrogliosis, and there is astrogliosis in this opposite way, and we could see that there is a decrease. I think that this is in the opposite way. So this is the these are changed. So this is the Huntington. The Huntington disease with astrogliosis, and this is the decrease after the treatment with the fingolimod. Also, we check the overactivation or NF-kappa B signal, and also that is found in the astrocytes, and also fingolimod could improve, well, attenuate this overactivation. What is interesting with this is that when we found that there is an increase after the treatment of BDNF, but we could not detect any changes in the BDNF protein. This happens several times because in the long-term treatments it's always difficult to catch where is the increase of BDNF. But we could see is that fingolimod treatment prevents the P75 upregulation in the hippocampus. You could see this, what we found in the beginning, that there is an increase of P75, and after the treatment, there is a reduction of P75. And also, this decrease that there is in the tract B, we could see also an increase after the treatment with fingolimod. So it seems that this drug is also modulating the P75 and tract B imbalance. We followed through trying to study the mechanism, and there is that this drug induces an increase in CMP levels and also promote CREP and raw phosphorylation that could explain the changes also in plasticity after the treatment. And another point is that this regulation of fingolimod could be induced by the modulation of TNF-alpha levels. When there is an increase in the TNF-alpha induces an increase in P75 that is reduced after the treatment. So in summary, this is the effects that we found. This was published very recently in the Human Molecular Genetics, and this was the cover of the issue that showed that fingolimod treatment can modulate the astrocytes, reducing the TNF-alpha, and also modulating the tract B and P75 imbalance. And also we were very happy that this was also chosen as a new in the Nature and Reviews in Neurology about the possibility that this is a drug that is already approved that could be used for the cognitive benefits in the patients with Huntington disease. So this is what we wanted to show you, that we have different molecules with different effects, and to try to find a trophic therapy maybe is not enough just to give BDNF. Also, we have to try to improve the P75 and tract B imbalance. They are the people that did this last work with the fingolimod, and these are the group in the CIBERNED with the different PIs, Pep Canals, Esther Perez, and Silvia Gines. And thank you very much.